Good day, everyone. My name is Shalene Resurrection, and this is Skylar Regina Maril, and today we're going to present our research study entitled Chemical Changes in the Coloration of Sechium edule leaves Immersed in Different Solutions.
Let's start with the introduction. Sechium edule, commonly classified as sayote, is part of the Cucurbitaceae family with squash, cucumber, and melon. It has prolific growth habits and commonly found growing on walls in backyards. Sayote is a plant of the tropics and subtropics that could grow as an annual crop in the temperate zone that can offer a growing season of at least 6 months. It grows best in areas where annual daytime temperatures are within the range of 19 to 30 degrees Celsius but can tolerate 12 to 40 degrees Celsius.
Moving on with the bleach. Bleach is a liquid or solid chemical that is used to eliminate the primary color or whiten materials. They are generally utilized for decontamination and cleaning for the reason that they eradicate or regulate a lot of types of bacteria, mildews, algae, molds, and viruses. Bleach is a chemical that can remove or lighten color, and it is commonly via oxidation. Bleach is known as a stable alkaline solution, which comprises a pH level of approximately 11 or 12.
Next is vinegar. Vinegar is a sour liquid that is formulated by fermenting any dilute alcoholic liquids into a liquid comprising acetic acid. It is known that vinegar is an acidic solution having a pH level lower than 7.
Moving on with carbonated drinks. Carbonated soft drinks is a class of non-alcoholic beverage. It usually contains a natural or artificial sweetening agent, carbon dioxide, edible acids, natural or artificial flavors, and sometimes juice. Carbonated drinks are estimated to contain 94% water. However, their pH scale reaches around 2.5 to 3.5 and is a highly acidic solution.
Lastly, water. Water is a substance made up of the chemical elements oxygen and hydrogen that exist in states such as liquid, solid, and gas, and considered as one of the most necessary and abundant compounds on Earth. Water is also recognized as one of the primary key to an individual's survival. The standard range of the pH value of drinking water is in between the pH value 6.5 and 8.5.
Moving on with the situational analysis. Color changes are naturally occurring in plants. A few chemicals can be involved in the color changes of plants, namely chlorophyll, carotenoids, anthocyanin, and tannins. However, some external factors like type of soil, amount of sunlight, and location can also be a factor that can affect color changes in plants. With that in mind, investigations regarding the other factors that can affect the color changes in plants is needed so that scientists and other individuals would have a better understanding regarding plant health.
As mentioned, various external factors can affect a plant's growth as well as its change in color. A few abiotic factors such as drought, stress, temperature, and exposure to ultraviolet radiation can affect flower color variation. However, human activities can also affect plants in a way, but these occurrences aren't studied enough. For instance, individuals who are taking care of plants often put solutions or substances that they claim to aid a plant's growth.
Lastly, plants are very important to human life. It provides essential materials for human survival, such as oxygen. It also provides habitat for many species of animals all over the world. With that in mind, understanding how the process of plant growth works and what factors affects it would be highly beneficial for many people. Knowing the different species of plants requires different conditions as well to grow would be highly beneficial for all. With that in mind, having further study to know what is compatible for each plant and what affects it would be very advantageous.
For the literature review, anthocyanins are organically occurring pigments of red and purple. This supplies an excellent color that is broadly applied in numerous productions. With that in mind, the objective of the first study is to examine the influences of pH and color stability to anthocyanins that is used in food dyes. The anthocyanins was effectively extracted by the researchers from a rose red flower using maceration method. The outcomes showed that anthocyanins are constant at a low pH level, and this would display the color red. On the other hand, high pH anthocyanin displayed a change in its coloration, turning it into color blue. After the investigation, the researchers concluded that anthocyanins can be utilized as food dye.
For the second literature, the procedure and how alterations in flower color as well as the occurrence or non-appearance of nectar impacts the robbers and pollinators of Alcamara. This precise phenomenon was studied by Quinn and Lee. This research displayed how alterations in color affects the behavior of pollinators and robbers. The data acquired from this investigation proposes that the alteration serves a dual purpose of directing pollinators to rewarding flowers as well as reducing robbery in the end of maximizing pollination from indisputable pollinators and conceivably robbers. According to the author, the conclusions for Alcamara may apply to other species that contains a comparable floral change. Nonetheless, idiosyncrasies instigated by the identity of the legitimate pollinator and the robbers are probable. The author also stated that studying the idiosyncrasies with El Camara may be the next stage to completely apprehending the importance of floral color changes to plant health.
Let's proceed to the research cup. The researchers in the first study were able to gather sufficient data to prove that anthocyanins in plants can be an effective food dye. They were able to explain in their study that pH is such a vital part in maintaining the color of the anthocyanin in plants. However, the researchers only focus on the colors of the anthocyanins when its pH level is in extreme and low level. The researchers failed to have an in-depth explanation to what color will be displayed if the pH level of the anthocyanins are at neutral level.
Moving on, the researcher of the second study was able to give the sufficient explanation regarding the occurrence or non-appearance of the nectar impacts to the robbers and pollinators of N. camara, as well as the other mentioned occurrences in the given study. However, the researcher failed to give sufficient explanations or observations regarding the importance of floral color change to plant health.
Now, this study aims to investigate the chemical changes in the coloration of Sesum edule leaves immersed in different solutions that can be answered by the following questions presented.
The hypothesis in the study is also presented.
The research design incorporated in this study is a one-group pretest post-test design wherein 5 cutout leaves are assigned for the 5 different solutions. Thus, the state of the coloration of the leaves is observed.
Moving on to the data gathering procedure. A. Chayote leaves preparation. The researchers in the study gathered chayote leaves outside their own homes. The leaves were washed with tap water to make sure that the leaves were clean. The study only needs a small portion of the leaves, so the researchers decided to cut 5 1 by 1 inches leaves, which will be used in the experimentation of the study.
For the preparation of all solutions that is needed to conduct this study were available in the researcher's house. These are bleach, vinegar, soda, rubbing alcohol, and water. While for the experimentation, 1 cup of each solution was poured inside each cup. The researchers attached a thread with label in each leaf which are used as its holder on the cups. Afterwards, each leaf was soaked in each solution in 7 days. In each day, the researchers capture or records the difference of the leaves in different solutions every 3:15 in the afternoon.
Moving on with the data analysis. The coloration of the chayote leaves were subjected to content analysis to see if a change occurred or not. To be specific, the researchers first observed the initial color state of each leaf sample submerged in 5 solutions. The initial state of each leaf after being subjected to the said solutions were recorded by the researchers through documentation. For 7 days, the researchers observed each sample. Every change that occurred to the sample were documented and interpreted by the researchers.
At the final day of observation, after obtaining all the needed data, the researchers analyzed the difference between the initial and final state of each sample. The interpretation for each sample were based on the researchers' visual observation, and the analysis used for the data obtained from the experimentation is descriptive analysis.
Moving on with the results and discussion. Results showed that the solutions caused various changes in the coloration of each leaf. For the sample submerged in bleach, major discoloration occurred, causing it to fully dissolve after a week of observation. Other solutions, like water on the other hand, had no impact on the sample's coloration, which indicates that water does not cause any reaction to the coloration of leaves. For the sample submerged in vinegar, browning in the leaf's color was observed. The leaves submerged in vinegar went through many changes compared to the other samples submerged in different solutions. However, the changes that occurred were still minimal and were still within the shades of brown.
The sample's color change submerged in soda was different from the others because the color change of the edges were different from the center portion of the leaf. This was shown in the final day of observation of the sample submerged in soda, where the leaf's overall color was light brown, however the edges of the leaf was a brighter yellowish brown. While the color change for the sample submerged in rubbing alcohol went through a similar color change to that sample soaked in the soda, however, the only difference between the two is the color change in the leaf soaked in rubbing alcohol is lightened and cause a minimal darkening of its edges, not turning it into a different color.
Moving on with the conclusion and recommendations. The final analysis evaluated a significant difference between the coloration of the chayote leaves before and after being subjected to the various solutions, which indicates that the null hypothesis given in the study is rejected. With that, the researchers of the study suggest that further investigations regarding this topic can still be conducted.
Due to the fact that the ongoing pandemic is preventing the researchers from being able to use lab equipment that could possibly give a deeper and much more in-depth results, The researchers would like to recommend using better equipment if possible, such as chromatography tester.
Regarding the data gathering tools, the researchers would prefer having a more professional equipment that can monitor the changes occurring in the samples at all times. Other factors might also might affect the possible outcomes of the study. A few of these factors are temperature. This factor must be noted because having a constant temperature all throughout the investigation might cause a different effect to the samples than that of what the researchers have found.
The plants from the different areas might have a different outcome when submerged in the solutions used in the study. Hence, having the investigations regarding the leaves from different areas can help understand if the effects of the solutions to the samples would be the same despite its different area of origin. Now, the studies that are presented were the references used while conducting the study. And that concludes our presentation.